Hi everyone, my name is Carmen Chan, and today I am presenting my project titled Evaluating Structure-Based and Ligand-Based Virtual Screening Methods for the Design of Bcl-xL Inhibitors. This project was conducted under the supervision of Dr. Shapira and using the molecular software ICM Pro by Molsoft. Virtual screening is a computational technique used in drug discovery. It acts as a filter when processing large libraries of compounds in order to identify lead compounds for a chosen biological target that can be tested in the laboratory. As such, virtual screening diminishes time and costs by discarding unfavorable compounds early in the drug discovery process. There are many types of filters used in virtual screening, which generally screen compounds based on specified criteria and provide a score. One of these methods is structure-based virtual screening, which I will refer to as SB. The most common technique in SB is molecular docking, which screens the library based on structural complementarity between a ligand and the target receptor by attempting to predict binding poses in the receptor pocket and providing a score based on how favorable the pose is. This requires a known receptor structure in 3D. Alternatively, when the receptor structure is unknown, a useful strategy is ligand-based virtual screening, which I will refer to as LB. A technique used in LB is pharmacophore filtering, which relies on known active compounds as a template. The physicochemical properties of the templates are called a pharmacophore and are shown in the mesh. The library is screened based on their similarity to the template's pharmacophore. Each of these methods have advantages in green and limitations in red. To illustrate, SB provides information about receptor-ligand interactions. However, it does not consider protein flexibility unless multiple receptor conformational states are available. In contrast, LB provides information about ligand features that confer improved biological activity. However, the template must be representative of active compounds to be valuable in virtual screening, avoiding template bias and overfitting, which is limited by the availability of active compounds for the given biological target. As such, combining SB and LB may be a valuable strategy to exploit their independent advantages and improve the outcomes of virtual screening. A target that may benefit from a combined virtual screening approach is B-cell lymphoma extra large, or Bcl-xL, as both target structure and active compounds are available. It is an anti-apoptotic member of the Bcl-2 family of proteins localized to the mitochondrial outer membrane. It is encoded by the Bcl2-like-1 gene, or Bcl2L1, and composed of 8 alpha helical regions, which encompass 4 BH domains. The BH1 to 3 domains form a large hydrophobic groove that can bind strongly to pro-apoptotic proteins such as BH3-only protein BIM. Consequently, the function of Bcl-xL is to sequester pro-apoptotic proteins, thereby preventing cells from undergoing apoptosis. Consistent with this mechanism, cancer cells have been found to upregulate anti-apoptotic proteins to protect against apoptosis and contribute to chemoresistance. In contrast, studies have also shown that cancer cells upregulate pro-apoptotic proteins due to increased cellular stress from their hyperproliferative state, suggesting that cancer cells are actually primed to undergo apoptosis relative to normal cells. This poses an opportunity to specifically target cancer cells by inhibiting anti-apoptotic proteins without damage to normal cells. Together, these findings suggest that inhibition of anti-apoptotic proteins, such as Bcl-xL, may be a promising anti-cancer therapeutic strategy by tipping the scale to apoptosis in already primed cancer cells. In particular, a class of drugs called BH3 mimetics mimic the binding action of BH3-only proteins and inhibit the function of anti-apoptotic proteins such as Bcl-xL. As a result, more BH3-only proteins are liberated and can activate apoptotic responses. Currently, Venetoclax by AbbVie and Genentech is the only BH3 mimetic approved by the FDA for the treatment of chronic lymphocytic leukemia or small lymphocytic lymphoma. It acts by specific inhibition of anti-apoptotic protein Bcl-2. In contrast, inhibition of Bcl-xL has proven to be difficult. Previous attempts, such as ABT-737 and navitoclax, demonstrated potent Bcl-xL activity in vitro and in vivo. However, development was limited due to adverse effects. To illustrate, inhibition of Bcl-xL induced platelet cell death, as platelets rely exclusively on Bcl-xL for survival. Additionally, ABT-737 was not orally bioavailable. Considering this, inhibition of Bcl-xL is mechanistically a promising strategy for anti-cancer therapy. However, there is a need for improved inhibitors with better oral bioavailability and attenuated adverse effects. Virtual screening may aid in the pursuit of these new inhibitors, as both Bcl-xL receptor structure and active compounds are available, making Bcl-xL an ideal candidate for virtual screening. As such, the primary objective of this project was to determine the virtual screening method best tailored to identify compounds that target Bcl-xL. As a sample application of this finding, the secondary objective was to conduct hit optimization. The primary objective was achieved by evaluating three virtual screening methods: an SB approach using molecular docking, an LB pharmacophore filtering approach using rapid isosteer discovery engine based on Molsoft's atomic property fields and ABT-737 as a template, and finally a sequential combined method involving pharmacophore filtering followed by molecular docking. Using a hit from the determined best screening strategy, hit optimization was conducted, guided by receptor-ligand interactions and structure-activity relationships to achieve the secondary objective. The design of the present project was separated into three stages: validating each screening method independently, comparing enrichment, and hit optimization. The techniques used at each stage will be described. Prior to investigating the primary objective, the predictive capacity of each method was evaluated independently. The screening methods employed output two key pieces of information: a prediction of the compound's binding pose and a score generated based on how well the compound meets the method's screening criteria. Using this information, the predictive validity of SB and LB were assessed using pose prediction and ability to rank active compounds by score. The first piece of information, binding pose, was used to assess pose prediction. 13 compounds with experimentally derived crystal structures available on the Protein Data Bank were obtained and screened by docking to Bcl-xL or by pharmacophore similarity to ABT-737 as the template. For each compound, the crystallographic or native pose and predicted pose were superimposed, and the distance between native and predicted was calculated as root mean square deviation, or RMSD, in angstroms. In theory, good pose prediction entails that the predicted pose is very similar to the native pose, resulting in an RMSD lower than 2 angstroms as a cutoff. Here, the predicted pose in yellow is superimposed on the native pose in blue for a sample compound. The SB predicted pose is illustrated in Figure 1a, and the LB predicted pose is illustrated in 1b. As demonstrated, both SB and LB were able to accurately predict the native pose with an RMSD below 2 angstroms. The pose prediction of SB and LB for all 13 compounds are summarized in Figure 2. Here, the x-axis illustrates screening method, and the y-axis illustrates RMSD in angstroms. SB predicted poses achieved a lower mean RMSD of 1.51 angstroms compared to LB predicted poses with a mean RMSD of 1.89 angstroms. In further detail, the percentage of the dataset with RMSD above or below 2 angstroms for each screening method is summarized in Figure 3. Here, the x-axis illustrates screening method and the y-axis illustrates percentage of dataset, where blue is the percentage of the dataset that achieved an RMSD below 2 angstroms, and yellow is the percentage of the dataset with an RMSD above 2 angstroms. As shown, approximately 70% of the dataset was predicted within 2 angstroms of the native pose for SB compared to 46% for LB. Together, results suggest that SB was able to achieve better pose prediction compared to LB. The second piece of information, score, was used to assess the ability of the method to accurately rank active compounds. 1,057 compounds with demonstrated Bcl-xL activity were obtained from the Chembl database and screened by docking to Bcl-xL or by pharmacophore similarity to ABT737 as the template. Scores from the respective screening method, docking score from SB, and pharmacophore similarity score from LB were obtained. In theory, compounds with better Bcl-xL activity should confer better docking or similarity scores, as they have been demonstrated to have activity with this target. In Figure 4a for SB and 4b for LB, the x-axis illustrates activity as P-Chembl, a standardized metric for activity, and the y-axis illustrates the score output for each method. For both SB and LB, activity was not predictive of score demonstrated by R-squared values of 0.02 and 0.24 respectively, suggesting that both SB and LB were unable to accurately rank active compounds. Interestingly, LB ranking performed substantially better than SB. To summarize, results from this stage suggest that both SB and LB achieved accurate pose prediction, but overall, SB performed better. Interestingly, both SB and LB were unable to accurately rank compounds. These results are unexpected because good pose prediction should confer accurate ranking. A wider diversity of compounds in the ranking dataset may have resulted in poorer pose prediction compared to the smaller controlled dataset used in pose prediction. Additionally, the dataset used for ranking may have been more biased toward the template used in LB, resulting in the better observed ranking by LB compared to SB. To improve this, a more diverse array of compounds may be used for pose prediction to obtain a more accurate picture of the predictive capabilities of SB and LB. However, this is limited by the availability of crystal structures. Despite this, both SB and LB demonstrated some predictive validity in pose prediction, and therefore a comparison of their performance was conducted in the subsequent stage. The performance of each screening method was compared to achieve the primary objective using enrichment as an indicator of performance. Enrichment for each screening method is determined by screening a large library of compounds with no known activity, called decoys, together with a few compounds with known activity. A method that performs well or has good enrichment should be able to distinguish the active compounds from the many decoy compounds. As previously described, active compounds should be screened first and ranked better by the scoring functions because they are known to interact with the target. Following screening, early-stage enrichment is calculated as the ratio of active compounds in the top subset versus the ratio of active compounds in the entire dataset. 3 virtual screening methods were compared: SB, LB using ABT737 as the template, and a sequential combined strategy involving LB followed by SB. The compound libraries used for each method are shown, with the decoy compounds in gray and the active compounds in green. For SB, a diverse lead-like compound library from Life Chemicals was used as decoy. For LB and combined, a compound library from Cambridge was used as decoy. Active compounds from the Chembl database with a p-Chembl greater than or equal to 6 were used as active compounds to determine enrichment for each screening method. The results are shown in Figure 5. The x-axis illustrates the stage of screening and the y-axis illustrates enrichment for each screening method, SB in blue, LB in gray, and combined in yellow. Results suggest that the combined method achieved the best enrichment at multiple stages of screening, while LB performed poorly. In the context of drug discovery, this indicates that using this combined method, only a small number of compounds need to be screened to obtain a large number of lead compounds because they will be screened earlier. Results suggest that a combined screening approach may be a better screening strategy compared to each of the methods alone. Consistent with poorer pose prediction by LB observed previously, enrichment also demonstrated that LB alone is a poor screening strategy for Bcl-xL. To demonstrate the application of this finding, a hit from the combined method, shown in Figure 6, was optimized, shown in Figure 7. Optimization was guided by analyzing Bcl-xL binding pocket architecture, interactions with binding pocket residues, and structure-activity relationships. To guide hit optimization, analysis of the Bcl-xL binding pocket architecture was conducted. As previously mentioned, the BH1 to 3 domains form the Bcl-xL binding pocket. It is a long and shallow hydrophobic groove composed of 4 hydrophobic pockets, P1 to P4. Using ICM Pocket Finder to identify ligand binding sites, P2 shown in green and P4 shown in blue make up a substantial amount of the groove's volume. Analysis of pocket interactions with existing Bcl-xL inhibitors demonstrated the importance of P2 and P4. Looking at only the receptor pocket, endogenous Bcl-xL ligand BIM, shown in green, simply sits in the large hydrophobic groove of Bcl-xL, but does not interact deeply with the groove's pockets. In contrast, experimental Bcl-xL inhibitor A-1155463 displaces phenylalanine 105, creating a deeper P2 that is not observed in endogenous interactions. Hydrophobic interactions with P2 are strengthened by hydrogen bonding with serine 106 and leucine 108. Similarly, previously mentioned Bcl-xL inhibitor ABT737 engages P2 much deeper than endogenous ligands by pushing phenylalanine 105 into P2 with its chlorophenyl moiety. This displacement opens up the binding groove more than what is usually required to accommodate endogenous ligands. In addition to this, ABT737 engages P4 through a hydrogen bond with glycine 138 and pi stacking interactions with tyrosine 195. The interactions demonstrated by both of these potent Bcl-xL inhibitors supports the importance of engaging P2 and P4 for Bcl-xL binding activity. Subsequently, interaction hotspots in the Bcl-xL binding pocket were analyzed to identify residues that may strengthen binding. To do this, hydrogen bond interactions observed in the top scoring results of the SB docking study previously described were analyzed. Since SB was demonstrated to perform better at generating binding poses, only results from SB were considered. The results are shown in Figure 8a, where the x-axis illustrates various binding pocket residues, and the y-axis illustrates percentage of the docked dataset that formed hydrogen bond interactions with these residues. Residues were considered hotspots if more than 10% of the dataset interacted with it. As such, glycine 138 and arginine 139 were considered interaction hotspots for Bcl-xL binding. Figure 8b shows this finding in the mesh representation of Bcl-xL, where glycine 138 is colored green and arginine 139 is colored blue. Aside from the receptor, structure-activity relationships of compounds with Bcl-xL activity from the Campbell database were also analyzed. In particular, matched pair analysis was used to identify structure-activity cliffs. In essence, small changes between compounds that result in large changes in activity were identified. A very common substitution observed was a chlorophenyl group which engages P2 in 3D. An example of this change is illustrated in Figure 9, where the yellow compound has a p-Campbell of 5.96, and addition of the chlorophenyl in the blue compound achieved a p-Campbell of 9.89. The chlorine atom likely better occupies P2 and strengthens hydrophobic interactions. As such, this finding provides further support for the importance of P2 for Bcl-xL binding. In contrast, results from matched pair analysis demonstrated that there is little variability in existing compounds on the side that engages P4 in 3D. Using these observations, hit optimization was conducted using the MolSoft ligand editor with a focus on creating P2 and P4 interactions. Figure 6 illustrates the chosen hit from the combined method. It was chosen for hit optimization because it was the top scoring compound from the most populated scaffold, with a reasonable docking score of -32.60. A summary of modifications in 2D are illustrated in Table 1. Each change is accompanied by a radial topological convolutional neural net or RTCNN score, which is a deep learning-based model generated by MolSoft. Simply put, the score represents how good the corresponding modification is, where lower scores are better. Overall, the score was improved from -27.34 in the original hit to -44.47 in the optimized hit. The modifications will be described in 3D. The original hit in the binding pocket mesh is shown. As circled in red, there are areas of unexplored space in P2 and P4 that can be exploited by creating extensions from the original hit guided by previous observations. Starting in P4, the addition of this group in blue achieves pi stacking interactions with tyrosine 195 and forms a hydrogen bond with glycine 138, an interaction hotspot previously identified. In P2, addition of the chlorine atom shown in green better occupies P2, similar to the chlorophenyl group shown in matched pair analysis. Finally, addition of the ether shown in pink better occupies the binding pocket. These changes help better occupy the Bcl-xL binding pocket, especially P2 and P4, and make stronger interactions with the pocket residues, such as interaction hotspots. Optimization of a hit obtained from the combined method was demonstrated. It should be noted that interactions with hotspot arginine 139 were not utilized due to the planarity of the core structure. However, hydrogen bonds with arginine 139 are a conserved interaction in all endogenous ligands of Bcl-xL, and some potent Bcl-xL inhibitors such as A-1155463. As such, future directions should explore how interactions with arginine 139 alter Bcl-xL binding. Additionally, Bcl-xL binding specificity was not a priority of this project during hit optimization. This may be rationalized as studies have demonstrated that pan-inhibition of anti-apoptotic members of the Bcl2 family of proteins may be necessary to overcome chemoresistance, in which there is compensatory upregulation of anti-apoptotic proteins. However, Bcl-xL specific inhibitors may be useful in certain types of cancer. Table 2 illustrates protein sequence alignment of anti-apoptotic proteins Bcl2 and Bcl-W, which have high sequence identity with Bcl-xL. The residues highlighted in the blue box, alanine 104, serine 145, and asparagine 197, are unique to Bcl-xL. These residues may be exploited to achieve Bcl-xL binding specificity in future studies. The present project demonstrated that combining SB and LB may be a beneficial approach for screening compounds that target Bcl-xL. Specifically, SB molecular docking and LB pharmacophore filtering were explored. Hits will not only be similar to the pharmacophore of active compounds, but also fit favorably in the Bcl-xL binding pocket. Optimization of a hit obtained from the combined method was also demonstrated, guided by receptor-ligand interactions and structure-activity relationships. Additionally, LB consistently performed poorly compared to SB or combined. This may be explained by the template utilized for LB, ABT737. During validation, LB was able to predict accurate poses for compounds that share the same scaffold, but performed very poorly for compounds with different scaffolds. An example is demonstrated in Figure 11a, which shows the LB predicted pose compared to the SB predicted pose of the same compound in Figure 11b. Consequently, although it is recommended that a large number of compounds should be used to generate a representative template and pharmacophore, a major limitation of this project was the inability to generate a reliable template using a diverse array of compounds with Bcl-xL activity to conduct LB, as demonstrated in Figure 11c. The effects of using a single compound as the template were evident when conducting LB. Molsoft's rapid isosteer discovery engine, by default, returns hits that have at least 50% similarity to the template pharmacophore. However, for this project, the threshold had to be lowered to 25% similarity to obtain hits, suggesting that the screen was likely accepting any compounds after the 25% threshold. Similarly, the effect of this template was observed in other LB methods. Although not explicitly described, a reasonable 3D QSAR model was generated using the same template shown in Figure 12. However, this is again limited by the generation of a reliable template. If a reliable template can be generated, other LB methods, including 3D QSAR, should be investigated for Bcl-xL. Overall, these issues may be explained by template overfitting. Since a single compound, ABT737, was used as the template, the criterion for similarity when superimposing was simply too strict, providing little flexibility for compounds with different scaffolds. This is a clear example of why it is necessary to strike a balance between a strict and a fuzzy or generalized template. When screening for new compounds, a strict template, as demonstrated in this project, may achieve better activity but may be susceptible to overfitting, as well as decreased structural diversity in the hits. In contrast, a more generalized template may allow increased structural diversity and scaffold hopping, but increases the chances of false positives with activity. Alternatively, P2 and P4 were demonstrated to be important for binding, and the chlorine atom in P2 greatly improved activity. If generating a reliable template with multiple compounds is limited, it may therefore be helpful to instead prioritize these features when generating the template, or increase the contributions of atoms in these regions, such that some strictness is retained while providing more flexibility in other regions. In conjunction with overfitting, the utility of LB in general for Bcl-xL may be limited. Using Lipinski's rule of 5 as an indicator of drug likeness, ABT737 breaks almost all the rules, with the exception of number of hydrogen bond donors. To explain this becomes a discussion about the druggability of the Bcl-xL binding pocket. The shallowness of the groove limits the number of surfaces to make interactions with, so the best way to achieve binding would be to extend lengthwise and exploit the surfaces that are available. As a result, compounds with Bcl-xL activity are very long and complex molecules, like ABT737. In the context of virtual screening, this complexity becomes a challenge, especially for LB methods. For example, the complexity of these compounds makes it difficult to identify hits that fulfill this just as complex pharmacophore, supporting the conclusion from this project that LB alone is not sufficient for Bcl-xL. This project did not address persisting issues previously described to limit the development of these inhibitors, such as limited oral bioavailability and thrombocytopenia. Future research efforts may improve a combined screening strategy by employing other types of filtering. For example, a recent study by Wen and colleagues utilized drug likeness filtering based on Lipinski's rule of 5 to screen for inhibitors of Bcl-2. Employing this considers bioavailability early in the drug development process. Additionally, Touré and colleagues described the heterocyclic replacement of the acyl sulfonamide core in ABT-737, which improved its solubility in vivo. Alternatively, Bcl-2 and Bcl-xL inhibitor AZD4320 and its dendrimer conjugated counterpart AZD0466 were recently demonstrated to induce transient thrombocytopenia, which recovered within a week. This finding suggested that drug-mediated thrombocytopenia may be attenuated by a weekly dosing schedule to allow platelet recovery between doses. Additionally, Kahn and colleagues recently described the development of DT2116, a proteolysis targeting chimera, or PROTAC, involving navitoclax linked to an E3 ligase ligand, thereby marking Bcl-xL for VHL-mediated proteolytic degradation. Since minimal VHL is expressed in platelets, the DT2116 PROTAC strategy protects against platelet cell death. This was demonstrated to have potent anti-tumor activity, greater than navitoclax, and less platelet toxicity in vitro and in vivo. Interestingly, in March 2021, DT2116 was approved by the FDA to move forward to clinical testing, suggesting promise in the future of targeting Bcl-xL activity as a therapeutic strategy. To conclude, findings from this project demonstrated that a combined approach involving both structure-based and ligand-based virtual screening methods may be beneficial for identifying new compounds that targets Bcl-xL compared to each method alone. Utilizing this approach may improve the identification of new anti-cancer therapeutics by upregulating pro-apoptotic and downregulating anti-apoptotic responses in cancer cells. Thank you to Dr. Shapira for showing us the ropes in ICM Pro and for his guidance throughout this project. Here are my references. And thank you for joining me today.